Um, wow, that's awesome. Thank you so much for the great introduction, and I'm so happy to be here and have this opportunity. Thanks also to the organizing committee. So, let's see. So the foundation for what I'd like to discuss here is this now very well-known idea to the fMRI community based on landmark studies from Bharat Biswal and Marcus Rakel and others. And this is that the brain is, of course, never truly at rest, and that most of the brain's energy consumption is in its intrinsic activity. So if we study only the externally evoked task activation, then that might just be the tip of the iceberg. And if we look at fluctuations in these ongoing signals in our fMRI data, they may appear to be random at first glance, and a long time ago they were just treated as noise. But we now know that they actually have beautiful organization in their correlation patterns and other signal properties, and then this can reveal large-scale distributed networks down to organization on much finer spatial scales. So studying these spontaneous fluctuations with fMRI has provided a powerful window into brain functional architecture. And now, of course, as you know from the exciting symposiums at this meeting, there have been many exciting lines of research that are trying to push the boundaries of what we can learn about the brain based on these signals. So the theme I wanted to center on today is going to be how can we obtain richer information from intrinsic fMRI signals to ultimately further advance neuroscience and clinical research. And there are many different paths to this question, and here I plan to touch on 4 different areas that we might push forward toward this goal. And I hope to illustrate that through these kinds of directions and through the work of many, many different research groups, it's become evident that these spontaneous fMRI signals are indeed so rich and that there's still a lot of promise for unlocking even more information. Oops, right. So to start, one of the most active endeavors in resting state fMRI has been in the development of new techniques for analyzing this kind of data. So this has revealed functional organization in exquisite detail, including network properties, boundaries between functional regions, as well as more continuous spatial gradients. And one thing I wanted to highlight is that most methods for analyzing intrinsic activity have primarily emphasized a spatial perspective. So what are the spatial patterns by which these signals are organizing and correlating with each other? And indeed, the most common way that we do fMRI analyses, both for task and resting state, has been to take all of the time points we have across the whole length of our scan, so whether that's 5 minutes or 15 minutes, and we use all of these time points to try to make a single inference or map of some metric of interest, like the correlations between different brain areas. And so this essentially provides us with a time average summary of our data, and this has certainly unlocked a lot of valuable insight. And it makes actually a lot of sense, since in fMRI we're usually more limited in the time domain compared to space. And also our data are noisy, so usually it helps to include as many time points as we can in order to boost our statistical power. But we can imagine that there may be other things to be found if we consider the time dimension more explicitly. So our data are inherently 4-dimensional, and when we look at it in this kind of a movie form, I think it can inspire a lot of questions about the more moment-to-moment variations in these signals, such as, can we learn more about how information may propagate between large-scale networks and how the brain is altered across important internal state changes like attention or motion? And if we can learn the temporal signatures of these internal states, and if these can provide us with sensitive indicators of disrupted brain dynamics. And so it was about 10 years ago, and a little earlier than that, that several studies began to come out that started to try to dig into these types of questions and try to see what more we could learn by more explicitly using this time domain. So for example, what happens if you start to divide the data into different windows of time points or different sections and look at the connectivity changes across time, as in this work from Vince Calhoun's group? Or are there repeating spatiotemporal patterns in the data, such as Sheila Keilholz's group has found, or modes with distinct temporal dynamics? And And instead of summarizing the relationship between two brain areas as just a single correlation coefficient, what if we open that up over time and look at how coherence might be changing across multiple timescales, and if that can possibly tell us anything meaningful about natural variations in behavioral or cognitive states? And Danny Bassett has also been doing brilliant work in looking how networks change during task and learning, also around this time. So in parallel to that, there are these efforts to start mining the temporal dynamics of the more endogenous resting state brain activity. And then, since then, more approaches have been introduced for this analysis, again, by many different groups, including methods like clustering, hidden Markov models, transient co-activation patterns. And there's also been more research on whether these temporal dynamics may provide valuable biomarkers of disease, such as by looking at the time that one's brain activity might dwell in a particular configuration. And at the same time, there's also been important dialogue about how we should best carry out both the analysis and the statistical testing if we're interested in studying time-varying effects, since it's actually quite easy to get spurious dynamic phenomena that come just from how we do the analysis, even if the characteristics of our data may not be changing. And so I believe that as we advance these approaches for looking at the data in time-resolved ways, it opens up new possibilities for capturing more information about brain dynamics and the internal processes that those may reflect. But at the moment, we might agree that we also face some gaps in being able to make full use of this data and to directly link the observations we see in our data to new knowledge about brain function. And part of this comes from the usual challenges that we face with fMRI, which is that we're looking at an indirect and slow hemodynamic measure. And so even if we move our analyses to much faster timescales, we're still far away from the millisecond temporal dynamics of a lot of neural processes. And actually, people have been looking at spontaneous brain dynamics with electrophysiological data for much longer than we have with fMRI. We're also looking at kind of small and noisy signal fluctuations. And another challenge that we have is that the signals we observe are a complicated mixture of diverse types of neural activity and state changes. And so, for example, if you imagine the activity we might see in some particular voxel, some of it may relate to thinking about something, like maybe planning your day, and that voxel is probably active as part of a larger functional network. But at the same time, there might also be variations in how alert you are, so that voxel signal might also be modulated by subcortical areas that relate to vigilance. And it's not straightforward to unravel these different contributions to isolate specific effects of interest. And so a broad question we can ask is, how do we go about accessing features of these signals that are linked to meaningful aspects of brain function and neural dynamics? And there are several complementary lines of work that could help to advance this. And as just a few examples, computational modeling can help to bridge large-scale patterns that we observe with faster timescale neural dynamics. And with the growing availability of big data sets, there's also been more opportunity to use techniques like machine learning or advanced statistical techniques to decompose fMRI data, understand its latent structure, and link certain fMRI features with phenotypic measures. And another one that I'm going to mainly focus on today is this idea of combining fMRI with complementary techniques for measuring intrinsic activity, as well as techniques for modulating neural activity in order to probe the role of different circuits in generating these signals. And so overall, hopefully, the more clearly we can ground our understanding of the fMRI signal changes, then the more powerfully we can use this data. So before discussing what kinds of neural dynamics might be in our data, one question we might ask is about which parts of these signals are probably not neural, so that we can try to distinguish those and improve the accuracy of our studies. And this is something that I became really interested in during my PhD research, though that was not the original plan I had in mind. So I was actually experimenting with seeing how functional connectivity might change over time, like what happens if you make correlation maps of the default mode network over different chunks of time. And one thing I had noticed was that sometimes there were windows that had a large amount of correlation across the brain. And since we were recording physiological data, like how the person is breathing over time, I could see that often it seemed to happen when the subject was taking a large, deep breath. And if we look at the raw signals, so these are time courses of each voxel in the brain lined up in this matrix representation introduced by Jonathan Power, it clarifies that the effect we saw may come from the fact that deep breaths cause a perturbation of the fMRI signals across widespread areas of the brain. But this was known already at the time. So studies by Rasmus Byrne and Richard Wise and others had shown that in addition to the more instantaneous artifacts we could get in fMRI with each breathing cycle, which perturbs the magnetic field, there are also slower natural changes in the depth and the rate of breathing, which also impact fMRI signals. So since BOLD is a hemodynamic signal, and since brain is, of course, connected to the rest of us, anything going on in the body that can modulate blood oxygenation in the brain is going to produce BOLD signals that are largely uncoupled from the local neural activity. And these respiratory variations are right in the same low frequency band as BOLD fMRI, and in fact were shown to correlate with intrinsic fMRI signals. And so for that reason, the attention to these kinds of effects increased together with the growth of a resting state fMRI field. And since this can influence the results of our analyses, one question is, how can we actually isolate the specific influence of physiological fluctuations in our data so that we can account for that and more accurately map neural activity and connectivity? So a number of different methods have been proposed for this. One approach that we and others have taken involves monitoring cardiac and respiratory activity during the scans and then trying to model how those effects propagate into fMRI time series. So there's not just a straight correlation between the two signals, but the transformation may be a little more complex. Rasmus Byrne had shown that the influence of taking a deep breath on the fMRI signal may be well modeled by convolving respiratory variations with a respiratory response function, which looked like this, analogous to how we convolve neural activity with a hemodynamic response function in order to try to capture the transformation between local neural activity and the BOLD response. And building on this idea, we replicated this to show a similar respiration response function during the resting state, and one for changes in heart rate as well, by deconvolving these from the resting state data. And so the overall goal here is that if we can construct models that link these known measured changes in physiology to fMRI signals, then we may be able to more clearly resolve and disentangle these in our data, and hopefully with less danger of removing a neural activity of interest than we might get with certain alternatives like global signal regression, which just removes a whole brain average signal in order to try to clean up the data. So global signal regression has been controversial because we know that it contains an unknown mixture of both artifacts and neural activity. Another way that physiological effects diverge from a simple whole brain average is that if we take a closer look, they're not simply uniform and global across the brain. So they may have interesting dynamics that differ across different brain regions. So for example, here if we try to deconvolve respiration or cardiac response functions for each voxel, and then cluster voxels that share similar response shapes, we can find that different sets of areas may show different physiological dynamics. And this was an earlier attempt, and more recently, Jingyuan Chen, who's working in John Palomeni's lab, used a large number of subjects from the Human Connectome project to show that there are clear and organized spatial patterns of areas that have distinct respiration responses. And these responses were also reliable across two groups of 95 subjects each, which are these solid and dotted lines here. And if you increase the number of clusters, so from 2 to like 4 or 10, we may be able to continue to resolve organized spatial patterns that differentiate in their respiration responses. And so one thing that these studies indicate is that the physiological influences on fMRI are more complex than we can explain with a single global regressor, but also that we may be able to robustly characterize its signatures if we wanted to decouple that from neural activity. So far, we've actually just been discussing physiological effects as if they're confounds or things we want to remove. But it's also worth considering that we may want to study these signals too, as they've been shown to carry a lot of valuable information about brain physiology and about vascular properties of different regions. So for example, The relative timing at which they impact the brain may signify pathological changes in the vasculature. And if responses to breathing are able to capture vascular hemodynamic response delays across many brain regions, this might also be helpful if we want to use fMRI to estimate neural timing differences, and perhaps if we want to model causal influences between regions in task or resting state. Another point is, although we often study the brain in isolation, there's been a lot of really intriguing work on this close and bidirectional connections between the brain and visceral signals, like from the heart or from the stomach, with our perception and emotion and cognition. And peripheral signals can also help to indicate different autonomic states. And so how can we best integrate physiological measures into our study of brain function, not only for cleaning up our signals, but also as meaningful sources of information? But going forward, I think there are a lot of challenges too if we want to build this complete picture of how physiological and fMRI signals relate to each other. And physiology is very complicated. The more I learn, the more I read, the more I realize how complicated it is. And it's been pointed out that there are nonlinearities or non-stationary behavior. So fixed linear models to relate them with fMRI may be too simple. There's also covariation between peripheral physiological responses and neural activity, which can make things difficult to disentangle. And then there have also been studies looking at different types of peripheral measures other than respiration and heart rate, and also seeing interesting dynamic patterns of propagation into bold fMRI signals. And so how then can we go about unifying all these different observations? So to summarize the section so far, the ability to distinguish physiological influences from more locally regulated, neuronally driven signals will better allow us to focus our investigation specifically on one component or the other without mixing up the two. And it's not always just about cleaning up noise, so it's becoming more and more clear that using our peripheral measures can help us learn more about brain physiology and brain-body connections. So now what about neural activity? Well, we know it's not all just vascular, so there's certainly evidence that intrinsic fMRI signals also relate to local neural activity, such as from more direct recordings in animals during fMRI. And in addition, the correlated network patterns that we see in fMRI data have also been found to be mirrored in the functional connectivity of electrophysiological signals in the human brain through intracranial recordings in epilepsy patients. And there's, for a long time now, been compelling evidence that the moment-to-moment intrinsic fluctuations have behavioral and cognitive relevance. So for instance, the pre-stimulus intrinsic amplitude and connectivity were found to relate to the way that an upcoming stimulus was processed. And there's also been studies using intermittent thought probes to try to query someone's mental state during the fMRI scan and relate that to the ongoing activity of different networks. Also, the characteristics of fMRI signals might also are known to show marked changes in characteristics across different state changes, such as arousal. And one finding, fairly robust finding now, has been that fMRI signals seem to increase in their fluctuation amplitude as people become more sleepy, and it also becomes more correlated across cortex. And consistent with this, Tom Liu's group had also shown that there was an inverse relationship between the amplitude of the fMRI global signal and someone's vigilance level. So, and here, vigilance was determined by simultaneously measuring scalp EEG during the fMRI scans. And actually, combining EEG together with fMRI has been a really valuable way of trying to probe the neural correlates of intrinsic fMRI signals. So EEG provides an established measure, a marker of different brain states, and it can let us clearly see electrophysiological indicators of state change, such as these fluctuations in the alpha rhythm, which could indicate levels of arousal. And here, by combining EEG and fMRI, we found that if we looked across the course of a 10-minute fMRI scan in a time-resolved way, windows of time where EEG indicated that the person was more likely alert came with different patterns of fMRI functional connectivity and signal amplitude compared to windows where the EEG showed that the person was likely more drowsy. So looking at these various results, one impression that we might take away is that everything is just so complicated. So sleepiness changes things, the way you breathe changes our signals, and what we think about changes our signals. And so how is it that we can possibly control for all these things? But what if we turn this around and actually see it as something that's very awesome? So if all of these state changes are showing up in our data, then it means we may be able to use them as a window into the dynamic internal state of a person and learn how brain activity may reconfigure across these varying states, both in the healthy brain and with different disorders. So, I think that with time-resolved analyses and through methods like multimodal imaging, this can be one approach for trying to draw that information out of our data relating to specific states. So in this context, I wanted to come back specifically to states of alertness, as this has been an important state change that has a close link with behavior and cognition. And in fMRI scans, it's easy to see a wide range of arousal states in our subjects. So some might be quite alert, maybe it's their first time in the scanner, but a lot of people actually fall asleep really easily. Now I have trouble finding people who don't fall asleep in my scans. And this could be a big confound if we want our subjects to maintain a well-controlled state while they're doing some task. But on the other hand, it provides us with this opportunity to study what happens in the brain as people are dynamically drifting through these states. And this was one of the earlier exciting demonstrations of the power to be able to detect changes in brain states directly from fMRI signals. And here they looked at sleep. And so EEG is the standard way of staging sleep. And here, by combining EEG with fMRI, they found that by taking short 1 or 2 minute windows of correlations between regions in the fMRI data, that this carried enough information to allow for classifying different sleep stages directly from the fMRI data, using a model that was trained based on the simultaneous EEG and fMRI data. But we also know that alertness can drift substantially even before we fall asleep. And the first time that I became very aware of this effect was when watching videos of monkeys falling asleep during resting state scans. So they actually seem to have the same experience as humans. It's a struggle to stay awake when you're in a dark scan or bore. And this was also something that I didn't intend to study at first. I was actually meaning to study, this was a very interesting data set where we could look at how invasive electrophysiology might relate to the fMRI. But once I saw these videos of the eye behavior that had been collected in this lab, I couldn't then unsee how large these state changes seem to be, and then realize that we definitely can't treat this as if it's one single resting state. And the first thing I did here was to just make a very simple regressor showing how open or closed are the monkey's eyes over time. And this is just to get a rough behavioral measure of how alert they might be. And then we can ask which areas across the brain were correlated with this in their fMRI signals. And what I saw actually surprised me at first, which is that most of the cortex seemed to negatively correlate with these behavioral alertness changes. And then that some focal subcortical areas were positively correlated, such as in the thalamus and also in the brainstem. But going back to the literature, actually this was not so surprising, as even the earliest simultaneous EEG fMRI studies in humans showed that fluctuations in the power of the alpha band correlate with fMRI data in this sort of pattern. And there have been a number of other studies to support this throughout the years as well, including work from Michael Chi's lab. And so, given that the way that fMRI signals may correlate with behavioral or electrical measures of arousal seem to be quite robust, we wondered if we could actually then train a model to go the other way around and do the reverse thing, or be able to leverage this type of pattern to be able to infer fluctuations in someone's alertness based on only the fMRI data alone. So to try this, we proposed a simple approach where we first learn a spatial template map that's linked with alertness, just like in the previous slides. And so we might learn this pattern from a set of training data, and then suppose you have an entirely new test data set that might not have measures of alertness like EEG. So we suggested that if we take this template pattern and then just take its running spatial projection with this data on a time frame by time frame basis, so like for each TR in the fMRI data, then this could trace out a time course that we could perhaps take to be an estimate of someone's relative alertness across that scan. So to see if it might be working, here's an example where we formed an estimated vigilance or alertness index in a data set where we had not yet looked at any of the recorded eye or electrical data. And we found that it actually does a pretty good job of telling when the monkey had indeed spontaneously opened or closed its eyes. And then just to make sure that this was not just dependent on the behavior of the monkey's eyes opening and closing, but more rather something closer to the underlying changes in the arousal state, we took sections of data where the monkey's eyes were closed for a long period of time, and then using the electrophysiological data, we found that the match between our fMRI estimate and this electrophysiological indicator of vigilance was about just as strong as during the periods when the monkeys were opening and closing their eyes. We also have some evidence that this might work in humans. So here it agreed reasonably well with simultaneously recorded EEG measures of vigilance. And here we saw preliminary evidence that it might also be predictive of reaction times in humans performing a working memory task. So it might account to some degree for the trial-to-trial variability in fMRI. So now we're trying to make improvements to this method, but the idea here is that if we can combine fMRI with other measures in order to try to delineate the fMRI signatures of states like alertness, then we might be able to then detect those using only fMRI and basically get more information from these intrinsic fMRI signals. Also, beyond alertness, there may be other types of dynamic processes that may be identified in fMRI data as well, such as affect, cognition, including memory. And there are also multiple different techniques that can be used to identify these besides recording other measures. So, for example, in these studies, they engaged the subject with particular tasks or stimuli and applied machine learning techniques to identify the associated fMRI signatures. And another growing area is the use of naturalistic stimuli and intersubject correlations, which may be valuable for being able to hone in specifically on stimulus-dependent processes. How much time do I have? I can't see it. Huh? 15. Thanks. Okay, so a few things I wanted to emphasize in this section are that our fMRI signals are very rich and they reflect many different underlying processes. And while this seems complicated and scary on the one hand, it also means that we have this window into dynamic brain states of different individuals and that we may be able to unmix and capture specific types of neural information in a variety of different ways. And then once we can access that information from the data, we can try to channel that into a more complete understanding of things like individual differences or disease states or other aspects of human brain function. So far, we've been looking at fMRI signals and trying to relate those to certain state changes. But what that work doesn't actually clarify here is how these fluctuations may arise, or which circuits or networks are actually involved in modulating or generating these patterns that we observe. So if we go back to arousal again as an example, we know that subcortical brain areas and the ascending reticular activating system play an important role in regulating arousal states and have widespread long-range connections across the brain. And so we might hypothesize that these subcortical regions may be involved in somehow shaping the fMRI signals that we observe across the cortex. But it's hard for us to get evidence for this using only fMRI data, so we don't really have a way of telling for sure. But it may be possible to use invasive procedures in animals, for example, where we can do stronger causal tests to try to probe the role of particular areas in impacting the patterns that we see in fMRI data. And so here I wanted to briefly discuss one study right now where we had the opportunity to disrupt activity in the nucleus basalis, which is a small but very important region in basal forebrain. It's the major source of cholinergic input and has many important functions that relate to attention and sleep-wake states, for example. And in these experiments, we were able to transiently inactivate this region and then conduct long resting state scans to examine what happens across the rest of the brain. And this was done in monkeys who were not anesthetized. So the main finding was that inactivating this region actually had widespread effects on fMRI signals, and in particular, components that were broadly shared across much of the brain. And so when we inactivated nucleus basalis in one hemisphere, so we only did one hemisphere at a time, we saw that global fluctuations were diminished across the hemisphere that was on the same ipsilateral side to the injection. But that if we then switched this hemisphere and inactivated the other side, then we switched the direction of the effect. And we could see an effect not only in these correlation maps, but also if we just pull out raw time courses themselves from the brain. And so if we extract the signal from symmetric cortical ROIs, like here in the temporal cortex, the signal amplitude was suppressed in the hemisphere that was ipsilateral to injection relative to the contralateral side, which then also switched when we did this injection in the other hemisphere. But another thing we noticed is that even though we've been talking about global signals, this inactivation may have some degree of spatial specificity too. And so there were certain areas, like in the hippocampus and the cingulate cortex, that didn't seem to be much affected by our manipulation. So there was less asymmetry in our maps. And I had done this analysis and showed my colleagues who know neuroanatomy much better than I do that this is what we saw in the data, and they thought this was wonderful and very interesting, because it turns out that the area that we did the inactivation in doesn't have projections to hippocampus and cingulate. And so in basal forebrain there are distinct nuclei that have different long-range projections, and the effects that we saw were quite well aligned with the anatomy. But next, if we wanted to really test the spatial specificity more rigorously, then one thing that we could do is go in and inactivate another subregion in basal forebrain and see if this alters fMRI signals in a spatially distinct way. And that was what we found here. So there was actually, when we did this, originally we had done CH4AL. If we do CH4AM subregion, then there was actually not much overlap between the fMRI effects that were induced by those different injection sites, and then also agreed with what might be predicted based on anatomy. So to also bring this back to state changes, one thing that was very clear from this data was that the effects were dependent on the arousal state of the monkey. So if I looked at sections of the data where the monkey seemed to be very alert, we actually didn't see very much impact of this manipulation at all. It was really only when the monkey started to appear drowsy and fall asleep that we started to see this effect in the hemispheric disparity in both the amplitude as well as the global signal correlations. And so overall, one conclusion of this work was that disrupting this subcortical brain area had quite substantial effects over distributed cortical fMRI signals. And this was done, in monkeys But one hope is that if this kind of work can translate to the human brain, then it might help us to learn what we might expect in human subjects if these subcortical brain areas are somehow disrupted, either by pathological processes, maybe in neurological disorders, or natural processes like aging, and so that we can start to see what consequence it might have on fMRI signals across larger parts of the brain. And finally, this work also highlights the important influence of this ascending arousal system on our picture of brain function, and joins recent evidence in human subjects that also supports this importance of neuromodulatory systems on brain dynamics and on cognitive function. So in summary, these are just 4 different areas that I think can help us to push the limits of what we can study with fMRI signals. And as we discussed, all of these involve many open challenges and questions. While the more that we dig into these signals, the more complicated they may seem. On the other hand, this means that these data are much more rich than we could have imagined maybe a long time ago, and that they contain not only valuable information about principles of functional organization in space, but also about the dynamic state of the brain. And then hopefully we can bring this kind of knowledge together to try to gain more complete picture of brain function. And there are techniques that we can use to unravel these different layers of information in our signals and to try to pinpoint the signatures of specific ongoing processes, such as alertness or cognition. And if we can better understand these internal processes, then we can then start to study more about the ways in which these different states may be spontaneously expressed over time in different individuals or different patient populations. And that techniques such as combining fMRI with methods for directly perturbing different brain areas might give us more fundamental understanding of the fMRI signals and patterns that we observe. So I'd like to acknowledge many people who have been my mentors and colleagues and friends and provided so much inspiration for the work that I saw here. Also, a quick plug for this effort, which is a great way to support women in neuroscience. And thanks a lot for your attention.